Genetic diseases can be categorized in three major groups: Mendelian disorders, complex disorders which are affected by multiple genes and environmental factors, and chromosomal abnormalities. The Austrian monk Gregor Mendel established many of the rules of heredity by crossbreeding of plants in his garden. Now we use them as autosomal dominant, autosomal recessive, and X-linked disorders. They are consequence of single gene mutation and transmitted through the generation. Sometimes we face with some disorders that are influenced by both genes and environmental factors in complex pattern, such as hypertension, diabetes, allergic, and autoimmune diseases. The chromosomal abnormalities, including change in the number or structures of chromosomes. In these cases, karyotyping is useful. Mendelian disorders are set in three groups: autosomal dominant, autosomal recessive, and X-linked. This table shows you examples of Mendelian inheritance diseases that I'm going to talk about some of them. And what do pleiotropy and genetic heterogeneity mean? Pleiotropy occurs when one gene influences two or more seemingly unrelated phenotypic traits. Some examples of pleiotropy are sickle cell anemia, phenylketonuria that I will talk about later, and schizophrenia. For example, beta-globin gene mutation in sickle cell anemia will cause producing abnormal shaped RBC, leading to RBC breakdown, anemia, blood clotting, and different symptoms such as heart attack, stroke, kidney damage, and splenic or liver infarction. Genetic heterogeneity occurs through the production of single phenotype by mutation at several genetic loci. One of the examples is retinitis pigmentosa, a group of genetic disorders that will cause loss of cells in retina. Mutation in one of more than 50 genes are involved. There is dark pigment deposits in the retina. As I said before, three patterns of Mendelian inheritances exist: autosomal dominant, autosomal recessive, and X-linked. Autosomal dominance is a pattern of inheritance characteristic of some genetic diseases. Autosomal means that the gene in question is located on non-sex chromosomes. Dominant means that a single copy of the associated mutation is enough to cause the disease. Both males and females can be affected and can transmit the disease. It occurs on non-sex chromosomes and at least one parent is affected. Clinical pictures can be modified by reduced penetrance and variable expressivity. I've talked about homologous chromosomes before. One homologous pair comes from mother, we call it maternal chromosome, and one comes from father, we call it paternal chromosome. And as I said before, these chromosomes are similar. Each carries the same genes in the same order, but are not identical. For example, the gene of eye color is located in the same area, but one of them carries the eye color comes from father and the other one, eye color of mother. In this picture, you can see homologous chromosomes in somatic cells, which one set comes from sperm and one set from egg. In autosomal dominant inheritance, just one mutated gene is enough to cause condition. In this picture, the normal gene is colored orange and the mutated gene is blue. In autosomal dominant inheritance, just one mutated gene is sufficient to cause condition. When one parent, father in this example, is affected, each child has one chance in two of having the disease. It is not sex-related and each parent can transmit the condition. Sometimes autosomal dominant disorders happen with normal parents. This occurs when new mutations involving egg or the sperm which they were derived. There is no increased risk for development of the disease in their siblings. Penetrance is the proportion of individuals carrying a particular gene that also express an associated phenotype. For example, disorder with 95% penetrance means 95% of Those with the mutation will develop the disease and 5% will not. Other gene suppressors and environmental factors can affect penetrance. Expressivity is the degree and the intensity of a given phenotype expressed by individuals having a particular genotype. For example, in neurofibromatosis type 1, the symptoms range from brownish spots on the skin to multiple tumors. Look at this slide. You can compare genetic penetrance and expressivity. Penetrance is the proportion of individuals with certain gene that also express the associated trait. You can see it in second line. The expression is the intensity of phenotype appearing by the gene. In third line, you can see variable expressivity by a variable intensity of color. Autosomal dominant mutation can affect membrane receptors, structural and transporter proteins. For appearing the clinical signs of altered gene, 50% reduction of gene product is necessary, but 50% reduction of enzyme activity can be compensated. Therefore, autosomal dominant disorders usually do not encode enzyme proteins. Recessive refers to a type of allele which will not be manifested unless both of these copies of the specific gene have the particular genotype. Contrasted with the dominant pattern, if one copy of the gene is present, the dominant allele will show itself. A carrier, an individual who carries the gene, is capable to passing on a mutation and may not display disease symptoms. In order to have the disease, an individual must have inherited mutated alleles from both parents. An individual having one normal allele and one mutated allele does not have the disease. The chance of producing child with disease is 25% by carrier parents. So, when both of the allele are mutants, recessive disease will become symptomatic. In this picture, the parents are carriers, means that each one carries one mutated gene. 50% of the children are carriers, and the risk of affected children is 25%. In autosomal dominant, I describe variable penetrance as the proportion of individuals carrying a particular gene that express an associated phenotype. It means that some individuals don't show associated phenotype even when they carry the gene. In autosomal recessive, complete penetrance is common, and the onset of disease is early in life. Enzymes are affected by this type of mutation because two copies of affected allele is present and no compensation is happened. Humans have two sex chromosomes, the X and the Y. In an X-linked disease, males are usually affected because they have a single copy of the X chromosome that carries the mutation. In females, the effect of the mutation may be masked by the second healthy copy of the X chromosome. As I said, male has one X and one Y sex chromosome. The X is transmitted by mother, so the female carriers transmit the mutated gene on X chromosome only to sons. In heterozygous female carrier, one X chromosome carries the mutated gene and the other X chromosome is normal. So when the father is unaffected, carrier mother transmits the mutated gene to half of offspring. Each son has 50% risk to get the mutated gene from the mother and become affected, and half of the daughters are carriers. An affected male transmits the X-linked related gene to all daughters.